What is up, YouTube? Today we're going to be talking about the Hardy Weinberg Equilibrium. Now first, let's cover some important terms for you to know. Gene pool refers to all of the genes and alleles within a population. I actually like to think about it as an actual pool, and you got all the different alleles in here. Now, even though a gene pool refers to the total collection of alleles for all different genes, in this video, when I refer to gene pool, I am only referring to the collection of all the alleles for one particular gene. Also, when I say gene, think of trait or phenotype. The second term is allele frequency. Allele frequency is the total number of times an allele occurs divided by the total number of alleles for that same gene. So in this example, we're looking at the G gene. We have the formula for the allele frequency right here. So why don't we use this to calculate the allele frequency for the small g allele? Here on the top it says total number of times an allele occurs. And in this case, we're talking about the small g allele. So let's count. 1, 2, 3, 4, 5, 6, 7, 8, 9. It looks like we got 9 small g's. Alright, so we also want the total number of alleles. Well, we don't have to individually count these. We have 9 individuals. Each individual has 2 alleles. So 2 times 9 is 18. So the total number of alleles for the G gene is 18. Now we got 9 over 18. If we calculate that, we get 0.5. So therefore, the allele frequency of small g is 0.5. That also translates to 50%, meaning that 50% of these alleles are small g alleles. Alright, now with that out of the way, let's actually talk about the Hardy Weinberg equilibrium. When you imagine a population of organisms, you would expect their gene pool to change over time, right? It doesn't have to be a significant change, but certain factors like migration, natural selection, or even mutations would cause a change in the gene pool. Even if these changes are small, there's still changes, and we actually call these small changes in the gene pool microevolution, which is small changes in allele frequency within a population. Now, let's say the gene pool in a population doesn't change at all. Absolutely no microevolution. What conditions would have to be met in order for evolution to not occur in a population? Well, first off, it has to be a large population size, which prevents genetic drift from happening. Second, we need no migrations, so no one is coming in and no one is going out, which also means no gene flow. Thirdly, we need no mutations to occur, so that means that no new alleles can be introduced to the gene pool. The fourth condition is random mating, so that means that there's no mating preferences between individuals. And last but not least, we got no natural selection, meaning that there's no favorable traits, so each trait is equally likely to be passed on. Now, when all 5 of these criteria are met, we call it the Hardy Weinberg equilibrium. That means that no evolution is happening at all. Sounds unrealistic, right? That's because it is. Now, the Hardy Weinberg equilibrium is very rare in the real world. In fact, it most probably doesn't occur in the real world because you can't guarantee no mutations in a population. But anyways, the Hardy Weinberg equilibrium is basically a mathematical way of saying, if nothing's changing, here's what the gene pool should look like. It acts as a null hypothesis, giving us a baseline to compare real-life populations to. Think of it as the control group of an experiment. Now, in nature, unfortunately, we don't have control groups to compare to real-life populations. But what you can do is grab the population, write down the Hardy-Weinberg equation for it, and after a certain amount of time, you can compare the new real-life population to the Hardy-Weinberg equation to see if evolution has occurred. Alright, let's talk about the actual equation now. It looks like this. P squared plus 2pq plus q squared equals 1. And P plus Q equals 1. Now let's break that down. P is the frequency of the dominant allele, and Q is the frequency of the recessive allele. So, if P plus Q equals 1, that just means the total frequency of all alleles is 100%. I mean, no crap, obviously, is 100%. Now, P squared is the frequency of homozygous dominant individuals. 2pq is the frequency of heterozygous individuals, and Q squared is the frequency of homozygous recessive individuals. Now, I know this looks like a whole lot of new information and you're probably already stressing out. Don't worry, they give you this and this on the formula sheet for the AP Bio exam. And so you don't really need to memorize this. And this is probably still too much information for your fried brain to comprehend. So really just think of it like this. P squared equals big A, 2pq equals big A, small a, and Q squared equals small a, small a. And in that case, you might as well just think of this like this. P equals big A and Q equals small a. Wow, so simple. I just dumbed it down for you. You're welcome. Now let's try a quick example. Let's say that 9% of a population is homozygous recessive. Using this information, let's find all the genotype frequencies. Now, how are we supposed to do that? Well, using this, we know that q squared equals 0.09. Now to find q, we just take the square root of 0.09, which is 0.3. We know that p plus q equals 1, and we just found q. So 1 minus 0.3 gives us 0.7, so p equals 0.7. Now let's plug these into the equation here. P squared equals 0.7 squared equals 0.49. That's big A, big A. 2pq equals 2 times 0.7 times 0.3, which equals 0.42, that's big A, small a. Q squared equals 0.03. We already know that. That's small a. So in this population, 49% are homozygous dominant, 42% are heterozygous, and 9% are homozygous recessive. And there you go. We just calculated the genotype frequencies using the Hardy Weinberg equation. So why should we even care about this? Well, scientists can use the Hardy Weinberg equation to measure evolution. If the real life frequencies don't match what the Hardy Weinberg equation predicts, then that means that at least one of the 5 conditions are being violated. So evolution is happening. It kind of serves as a detective tool for genetic change. Alright, so now you have a solid understanding of the Hardy Weinberg equilibrium and how to use the equation. I hope this video helped, and thanks for watching.